There we go. Okay. Good morning, everyone. I want to welcome you. Good morning or good afternoon or good evening, depending upon where you're coming from. I want to welcome you to our virtual seminar series at the University of Utah Centers of Excellence and Molecular Hematology Center. Today, we have the pleasure of hearing from Dr. Jan Abkowitz. Jan got her undergraduate degree and her MD at Harvard and did her residency at Beth Israel Hospital in Boston. She then went to the University of Washington for a fellowship in Heme. And it's at during that fellowship, I think, maybe Jan could speak better to this, where her interest in research and red cell development became apparent. She made seminal discoveries in red cell biology, including the discovery of the gene FLVCR as a heme transporter. And she continues on in her research. She's had many leadership roles, including being the president of ASH. She participates in NIH study sections. I'm trying to remember, Jan, when we got introduced, but I'm sure it was through Jerry Kaplan's, and it's been a really great interaction with Jan and her research group. And we want to welcome her today for this presentation. The title of her talk is Heme Trafficking, Use, and Toxicity During Red Cell Differentiation. I will monitor questions in the chat. I'm going to ask everybody to please mute themselves except for Jan. And just remind you that if you want to ask a question, you can populate it in the chat, and we can I will sort of moderate to figure out if we want to populate those in the middle of the talk or at the end. Finally, at the very end of this presentation, I'm running a little bit of a poll. So if you have time to stay on at the end, I'd love you to participate. We're trying to figure out if this forum is a good forum that we want to continue through the next year. And also let you know that if you are interested in presenting, to please reach out to me. So that I can sort of get a list of those individuals who might want to participate in presentations in the future. And with that, I will let Jan take over. Thank you, Jan, again, for your presentation today.
Yeah, thanks, Lois, Diane. It's my pleasure to talk to everyone today. And I think this has been a wonderful forum. How often do we have such an international crowd at all hours of the day being able to talk about iron? It's just been tremendous. So I'm going to speak about heme trafficking, use, and toxicity during red cell differentiation. And I don't think I have control here, and I'm not sure why. Here we go.
Okay.
Okay. And what my plan is to first present some studies of MDS, of myelodysplastic syndrome, that result from isolated deletion of chromosome 5, called del5 MDS, and of a congenital anemia, Diamond-Blackfan anemia, probably known to many in this group, and show that these macrocytic anemias, macrocytic meaning big red cells, result from excess heme, and that understanding heme trafficking might identify new targets for therapy. And then I'm going to spend some time on unpublished studies from my lab for the last couple of years, where we use single cell technologies and combine them to look at molecular events that control both effective and ineffective red cell differentiation, and particularly the role of heme in these processes. So just as background, realizing when speaking to a red cell group, but also an iron and heme group, and sometimes we've been a little disparate as opposed to a single group, I thought I'd go through a little bit about erythropoiesis. And the most important message about erythropoiesis is that it has very rapid kinetics, that humans produce 2.3 million cells a second, and one can increase this 5 to 10-fold in response to anemia and is reflected in reticulocyte count. Hemoglobin comprises over 95% of red cell protein content. So the goal in life of a red cell is to become a nucleate sac, which contains hemoglobin. And this contains a lot of heme. And so large quantities of heme are needed, and it's needed very quickly as red cells mature. But the problem is that free heme is toxic, leading to lipid peroxidation, to high ROS, to cell death. So this is the dilemma that each red cell needs to face. That heme is a chemical chelate, and it's assembled enzymatically, and globin is a protein that's produced by transcription and translation. So co-regulation of heme with globin can't rely on the coordinate regulation of transcription and translation, and erythroid cells need an alternative solution. And that's what my lab has actually been looking at, the molecular coordination of human globin for many years. I'm going to present the solution and then some of its impact. So shown across the top are the stages of erythroid differentiation, beginning with the BFUE, the first committed cell, onto the blood where there's a reticulocyte and a red cell. And under the influence of GATA1, the erythropoietin receptor is upregulated. Erythropoietin comes, and then transferrin receptor is upregulated, transcriptionally regulated as well by GATA1. And so you have transferrin receptor expression on cells, and iron comes in. And then iron binds IRPs and induces ALO2S translation, which is an erythroid-specific enzyme. That's the first and rate-limiting step of heme synthesis. And so heme synthesis starts. And then heme induces globin transcription and translation by inhibiting BOC1 and HRI, and globin synthesis intensifies. And since BOC1 and HRI are repressors, globin synthesis starts rather quickly as soon as they're inhibited. But still, there's a period of time where heme exceeds globin in any normal red cell's life as it matures. And that's where we got into the picture by inadvertently cloning FLVCR, which is a protein which exports excess heme from the cytoplasm and protects CFUE and proerythroblast from heme toxicities. So it's absolutely needed at the proerythroblast CFUE stage of differentiation. And one can track these stages with markers, and traditionally they're tracked with CD71, the transferrin receptor, and glycophorin A, CD235. But for our purposes and many others, including Mohan and others in the field, the earlier cells are tracked with CD36. And for us, that's particularly important because iron output is a variable. So we didn't want to have that part of the tracking system. So I'm going to be looking at cells that have CD36, don't have glycophorin A, have both, have just glycophorin A, which is a traditional way of looking at cell differentiation. So just a little bit of background on the physiology of DBA and MDS with chromosome 5q deletion. Diamond-Blackfan anemia is a congenital anemia, congenital macrocytic anemia, and 25% of cases results from haploinsufficiency of ribosomal protein S19, and 65% from haploinsufficiencies of 19 other ribosomal proteins. So 20 different ribosomal proteins result in the same phenotype. And in addition, macrocytic anemia of del5q MDS results from the somatic or acquired mutation and haploinsufficiency of RPS14, making number 21. And when tested, these mutations disrupt ribosome assembly and impair protein translation. So since DBA and the anemia and del5q MDS share a clinical phenotype, we thought that slowed or impaired translation might be key to their shared pathogenesis. So here's the hypothesis that I'm going to be showing some data for initially, that heme synthesis intensifies, shown as the red arrow. This causes globin synthesis to intensify, that there's a short period of time where there's a gap and FLVCR is needed to export there. But in the setting of poor ribosome assembly that impairs globin translation, that gap can be longer. And if that gap is longer, then there's excess heme that extends for a longer period of time, and heme export via FLVCR might be insufficient. So the cell death occurs at this stage. And we think that this is a major contributor to the ineffective erythropoiesis in many processes that I'll allude to ahead. And a critical step, a critical obstacle really, that a single cell has to overcome as it differentiates and becomes a mature red cell. It has to handle heme relative to globin at this stage of its differentiation. So in some published data, we looked at a DBA patient, or DBA patients, 3 and 6 with L5-MDS, some normal individuals and some MDS controls, and looked at their differentiation in vitro. And this is just marrow directly from this DBA patient. What's shown here is actually a patient who's 28 and has RPS 19 haploinsufficiency and is transfusion dependent. So there's no confounding variable. And shown in red, one can see that she has a good number of early progenitor cells, same as the normal. But if one looks at those that are CD71 positive, express the transferrin receptor, there's very few in her, and they're blocked, while the normal would have this differentiation pattern. So she's blocked at the stage that we're interested in, where her cells don't readily progress past that. So we then went and used cell culture using the methods that are below by Mohan and group and others to look at how red cells matured in vitro. And looking at day 10 in our culture, normal maturation would go 1, 2, 3, 4. And in an example DBA patient, it doesn't quite complete that maturation and it dies off early. And if one looks at actually cell expansion in this example, that in stage 1, relatively same numbers of cells. This would be the BFUE stage. At the CFUE proerythroblast stage, there's a drop-off because of the cells dying. And the more interesting thing I'm going to tell you about is that those cells that appear to get through, that for whatever reason are able to adapt and become stage 2 cells, grow in number. So what we thought we were looking at was an obstacle at the CFUE proerythroblast stage that was induced by heme. And so to show that that was the case, we looked at it in very different ways. Shown here is the fold cell expansion, less as what I showed. If one looks at stage 1 and says, why in stage 1 were cells dying? One correlate is they actually had extra heme content. And then if one looked at stages 4 and 5, looked at the end, 3, 4, and say, who's allowed to survive? What selective advantage they have? Those cells in DBA that survived were those cells that either had less expression of ALS2 or more expression of FLVCR. So less ability to make heme or more ability to export heme. And if one looks at del5 MDS, it's the same. And actually those cells that survive have less ability to make heme, or really almost tenfold more ability to export heme. Now, those are great ways, but we were really proud of a different and third way of confirming these data. And that's looking at sampling marrow cell cultures over time. And why this is important, so the cells grow off over 17 days in this culture. But why this is important is that ALAS2 is an erythroid-specific enzyme, and globin is the erythroid-specific protein. So if we tracked ALAS2, which in this case we tracked the protein level, and beta globin in the same culture, then we have already internally controlled for any non-synchronous growth. We've controlled for different cell contaminations. It's not a pure culture. And we could look at just what's happening to erythroid cells over time. And so if one looks in this example at a DBA patient at day 3 in culture, there is plenty of ALS2 protein. Heme synthesis was initiating just fine. It was actually more than our normal control. But at day 10, there's very little globin synthesis relative to the globin synthesis in normal, meaning there's a very big gap between heme and globin. And so then we asked, what's going on in the middle, at the time that heme is in excess of globin in our cultures? And in the DBA culture, there is more ROS and there's more apoptosis. The same thing is true again with the del5Q MDS shown here, same experimental design. And then lastly, if one inhibits heme synthesis by using succinylacetone, which inhibits the second step of heme synthesis, that normal erythroid growth is slightly decreased, but DBA and del5Q MDS erythroid growth nearly doubles. So by inhibiting heme, one improves the differentiation of these cells. So as a first summary, our data showed that anemia occurs when the synthesis of heme in CFUE proerythroblasts exceeds its use in hemoglobin or its export via FLVCR. And by extension, limiting heme synthesis or restricting iron might be a novel way to treat DBA and del5Q MDS. And lastly, since diverse mutations in MDS could impede risk protein transcription or translation, and hence quick initiation of globin production, heme toxicity might also contribute to the anemia in MDS patients without del5Q, or an ineffective erythropoiesis more broadly. And this led to a unfortunately slowed to begin and recruit clinical trial, but with my colleagues, which was a staged single center open label phase 2 study of a chelator for persons with low intermediate 1 or low or very low MDS and higher ferritin, so iron accumulation, with the endpoint of whether they would improve their erythroid hematologic development. Now I'm going to turn for a minute to mice, because mice that are deleted of FLVCR have a macrocytic anemia that's similar to DBA and del(5q) MDS in its phenotype. As shown here, they're severely anemic, low hemoglobin, low hematocrit, and they have a high MCV, or they're macrocytic. And when one looks at mice where TR119 is the equivalent of a glycophorin A, and we use CD71 because they do not express CD36, and the same differentiation pattern can be seen in the FLVCR deleted, then in DBA, and in wild type there's a comparator. And you can see that there's essentially a block at the stage 1 in mouse where the cells accumulate in the FLVCR deleted animal. And if one looks at the stages in heme content, they're high. And if one looks at cytoplasmic ROS, it's high. And so we recognize that we had a reasonable model, mostly of what heme does in erythropoiesis. So now I'm going back to the same slide, and we're talking about mice. And CFUE becomes CFUE, the transferrin receptors upregulated, iron comes in, heme synthesis, the red line intensifies, globin synthesis intensifies as a reaction to that. Later in differentiation, TR119, the glycophorin A equivalent, is expressed. And there's a period of time when heme exceeds globin. But the problem in these mice is they can't handle this period that's a normal period of time. And so they die at this stage. And we recognize that there's nothing else wrong with these mice, that all of early erythropoiesis is normal. Everything else about their macrophage, everything else about their functions are normal. We've studied this quite extensively. And so it became a very interesting way to say, what does heme do at exactly this stage? Because it's when heme is expressed that they have a problem, they have to handle it. How do they handle it, and what is its consequence? And so since we've been talking about what's happening to a single cell, each cell, not a stage of cells with external interests, but an intrinsic property of every cell, we turned a number of years ago to looking at single cell experiments. And from now, I'm going to show you some data from single cell experiments in mice and our more recent data in single cell experiments in people. So the first study design was to identify early progenitors with flow cytometry that were essentially BFUE through basophilic erythroblast, to isolate single cells and quantitate each cell's cell surface marker expression, then barcode and sequence the cell's transcriptome. And what's unique to all the studies that I'm going to show is they contain normal ways that hematologists will look at cell surface marker expression as a way to document stage differentiation, and then the ability to link that protein expression to the total transcriptome. So just as an example that these methods are quite reliable, alpha and beta globin are transcribed on different chromosomes. And if the readout was reliable, there should be an excellent correlation between alpha and beta globin transcript. And indeed, as shown on the top part, there was. And if one looks at the correlation between TR119 and the glycophorin A expression, so gene and protein, there should be a good correlation, but not perfect. And similarly, if one looks at the relationship between the gene for transferrin receptor and the protein measured by CD71, the correlate should be good, but not perfect. As shown below, those correlations are quite excellent and not perfect. So the next thing we did is took a very unbiased method, which is a density plot. We recognized that TR119 was changing over differentiation. It's been well described. We said if you just, in an unbiased fashion, looked at the TR119 intensity and distributed cells based on that, rather than arbitrary criteria called TR119 high, TR119 low, which has been the way that it has been done before, that actually we identified 4 different TR119 groups that by density criteria were separate. And they're shown here in the control. We call them A, the most immature, B, the next, C, and then D. And then what we did in every mouse that we dealt with is we used these exact cutoffs that defined by this, and then said, how did this mouse distribute across those cutoffs. So here's the FLVCR deleted mouse. It's A, B, C, D. And we were very, very relieved when we saw that A had really two components to it, cells at this stage, some that had lots of transcripts in them that were alive, and some that had very few transcripts, which were essentially dying. And so about 50% of cells were dying at the A stage, which is exactly what we saw in our other studies of the mouse and what we would expect since this reflects the early CFUE stage. The second thing that one does typically with single cell experimentation is to look at what's called principal component analysis. And this plots 12,000 genes in terms of which cells are more similar, and then looks how they scatter, and shown here in 2 dimensions. And if you look at the early cells, the stage A cells, they overlap between the FLVCR deleted, called D here, and the wild type called W. However, if you look at differentiation, the wild type goes this way, and the deleted goes this way. And so this has meant to us that we would be able to find significant differences during the differentiation sequence over the CFUE proerythroblast stage that would tell us what's happening in these cells. And so the next thing one does is uses the gene sets. And this is, I think, of the order of 30,000 gene sets, and says, with all these gene sets, what are the differences that are significant? And to our surprise, the most significant difference between the FLVCR deleted and control cells were that the FLVCR deleted, and particularly in the early stages, A and B, had very high levels of RNA encoding ribosomal proteins. And then we thought about it and said, this makes sense. If heme is going to induce globin transcription and globin translation, it better well have ribosomes to be able to do the translation for it. And so this was something that was linked. And of course, we wanted to show that it was true. And to show it was true, we went to human. And so these are normal human CD36 positive, collective RNA negative, so sort of A and B kind of cells, earlier cells in human. And with the experimental design is at time 0, we gave these cells that were sitting just in culture. And so gave them ALA, and then said, what happened? Because what ALA does is it bypasses ALS2, so it bypasses the rate limiting first step of heme synthesis and immediately starts increasing heme synthesis in these cells. And then we look 15 minutes later. And 15 minutes later, after giving ALA, RPS14 message increased, and then 30 minutes later, its protein was up. The same was true with message and protein, both increased with RPS19 15 minutes later. And along with probably 2/3 of the RPS known small and large proteins, the increase was discernible with this experimental design. So in normal human cells with a 15-minute interval, heme, when induced to be synthesized, turns on the synthesis of ribosomal proteins.
Jan?
Yeah.
There's a question in the chat from Diane Krause, which is, what are the thoughts regarding why there is microcytic anemia in these patients and in the mice with FLVCR knockout?
Macrocytic anemia.
She's got typed in microcytic, but macro, yeah.
Macrocytic. Diane, let me get to that because that's going to be, I'll talk about that as I go along. Obviously, this is a linking format, a linking finding for those of us who deal with human conditions. And I'm going to make the argument that probably most or many macrocytic anemias are because of a hemoglobin disconnect. And I'll show you some of the reasons why as we go along.
Perfect. Thanks so much.
Yeah, thanks. And feel free to interrupt. That's super. So just to show that there's actually more ribosomes as well, we looked in the same model system at ribosomal RNA as a surrogate for ribosomal content. And within 30 minutes of adding ALA at time 0, there is more ribosomal content. The second most interesting thing was among the top 4, I think it was, most decreased messages in the FLVCR versus the control with all these gene sets was GATA1. And GATA1 had a very interesting pattern to it. And again, it was a little bit of a surprise initially that here is the control, and the control here for the GATA1 cluster genes, and this is the deleted. And if one looks in the A group, so at the beginning, they both are pretty low. But if you go to B, C, D, it increases significantly in the control, and it's very minimal in the FLVCR deleted. And one can look at this in different ways, a gene cluster analysis, looking at the genes that Emery Resnick, I don't know, Emery, if you're on the call or not, but you are very helpful to us with this gene set, showing us ones that have been known to be gene responsive, GATA1 responsive, and A is similar in both, but there's a lot in the wild type of upregulation of genes that are GATA1 responsive and not in the FLVCR deleted. And we did the same experimental design with adding ALA at time 0 to normal human cells, and rather quickly, there is a decrease in GATA1 within 15 minutes, shown both at the protein and RNA level here.
One more question in the chat. Sorry for the interruption.
Oh, I'm happy for it.
Okay, that's what I thought, so I'm going to go for it. Stefano Ravella is asking, is there any relationship between transcription factor for ribosomal genes and heme as a cofactor?
Yeah, I have no idea. I think that's a great area to, Stefano, to look at. We're actually asked by the reviewers to look at ribosome content to complete the message. And so this was an add-on experiment to which we're very pleased for that suggestion. And we haven't really pursued it any further. So I don't know anything. We don't know a lot about what, how or why heme transcribes certain messages other than the BAC1 connection. It's just not an area that we've really explored.
Thank you.
So as a last set in this publication, we looked at K562 cells, so human erythroid cells, and did an interesting experiment. And the intent of this experiment was to show that it was specifically heme that was driving the abnormalities that we saw. And so in the red, we added ALA, the same study design that I showed before. And at time 0, the content of heme is 1. At time 15 minutes, it increases. At time 15 minutes, ROS increases. At time 15 minutes, GATA1 protein is down. And at time 15 minutes, globin protein is actually already increasing. And then we did the important control, which we added simultaneously ALA to start the first step of heme synthesis and succinyl acetone to block the second. So we started it in the same way, but we didn't let it happen. And then if heme synthesis doesn't happen, there's no increase in heme content, there's no increase in ROS, there's no decrease in GATA protein, and there's no increase in globin protein. So we thought this was pretty specific that heme was inducing these changes that we saw in the different systems, mouse, cells, cell lines. And this led us to think this way about what's happening with heme and GATA1 during normal and aberrant, meaning del5Q and MDS and FLVCR differentiation. So here's again the scheme of differentiation. And GATA1 upregulates the transferrin receptor and ALS2 transcription starts the program. And heme synthesis intensifies, which upregulates globin synthesis and ribosomal protein transcription. That over time, as red cells mature, their FLVCR surface expression decreases. And this is because it's only synthesized before the CFUE stage, and then it dilutes as cells divide. So by the late stage, FLVCR is very low, and heme again accumulates. And this might assure that GATA1 is downregulated and that erythropoiesis actually ends appropriately. And the work of others, if one overexpresses GATA1, there's failure to erythropoiesis at this stage, it doesn't end appropriately. So what then would be happening in del5Q MDS and DBA and FLVCR deficient erythropoiesis is that heme accumulates early because there's premature downregulation of GATA1. So heme accumulates early and there's premature downregulation of GATA1. And so erythropoiesis prematurely terminates just at the stage that we're talking about. And so in summary too, looking at what we think is the physiology of these two diseases, genetic mutations in MDS directly or indirectly result in slowed transcription or translation. In DBA, translation is slowed. And in all these circumstances, heme exceeds globin. Again, heme being a chelate, so made by enzymatic process, so you need very little protein, and globin being a protein that you need in gobs. So there's a major stoichiometry disconnect. And in a situation where you can't make protein, heme's going to exceed globin. The excess intracellular heme causes increased ROS and cell death. It increases ribosomal protein synthesis, which in the setting of the, if there's which should amplify a ribosomal protein imbalance if present, so in the setting of the two diseases, and cause more nucleolar stress. It also decreases, and this is Diane, the transcription of genes involved in mitosis, which may be one of the contributors to macrocytosis. And I'll show you that data in a murine model of DBA in a minute. But it's also true in the FLVCR deleted mouse. And it decreases GATA1, resulting in earlier premature termination of the erythroid differentiation program.
Have another question in the chat.
Yep.
It's from Oleg Kalamanchuk. Has anyone tried to dissect heme and ROS in the experiments you've presented by antioxidant pretreatment?
Yeah. I'm not sure that that's going to be a sufficient way to dissect them. And we really haven't. There's others that have, I guess the best answer is no.
That's
going a long discussion of sort of tangential ways in others' work that that have tried. I kind of think of them as shown here, that excess free heme causes ROS, and so you're seeing the downstream consequences of both, where ROS has its own things that it's important for transcriptional regulation. And I would say that's not that well described, because ROS is just an entity, it's this phenomenon, and heme is actually a product. And I don't know exactly how to separate the two. So I think of it just as shown here, that excess free heme causes ROS, and either the free heme is a transcriptional translational driver or undriver, or the ROS leads to ferroptosis and apoptosis and other downstream consequences, and just a cell demise. And I really haven't explored that. And when there's poor ribosomal assembly, for example, in DBA, then there's also nucleolar stress, which activates p53 and causes another direction. And what I want to point out for the clinicians that are here is that the different ways that we approach patients with these disorders imply that if we were able to decrease heme synthesis, that it should be synergistic or at least additive. And that's because all of the approved therapies essentially lead to either more erythroid cells, or in the unique situation of del(5q) MDS, to less p53 activation. And so theoretically, one could have additional gain by combining erythropoietin and decrease of heme synthesis, or altrombopag or polymerase inhibitor, along with a decrease in heme synthesis. And these are mostly either used or in progress drugs that are listed on the right.
Two more questions in the chat that I'll put here.
No, it's okay. Keep going.
Okay. Is there any evidence that FLVCR deficient mice have suppressed HRI activity to drive globin translation? In other words, will you expect HRI deletion may partially correct the phenotype of the FLVCR?
Have not done that. It's actually a great study, and I don't know. I think that that's downstream of FLVCR, and so there's erythropoiesis is going to have challenges at very different levels, and I think that's an interesting study, and we have not done it. I'm going to end with a little bit of a comment about big and little cells, and because it's very well known that. From the work of Jane Chen, that HRI is important for controlling cell size. And this gets back to Diane's first question. So let's save that a little bit. But the real answer is I don't know.
Okay.
So shown here is essentially the same studies done in what we think is the best DBA mouse model, which is an RPL11 haploinsufficient mouse. So it's a mirroring DBA model. The findings are similar to FLVCR deleted mice. And shown here. So here's control, here's FLVCR deleted, here's RPL11 hets. And this is a, oops, I'm labeled, yeah, it's up top, very small. These are ribosomes that are increased in the early erythropoiesis. This is GATA1 cluster that's decreased in late. And relative to Diane's question, in both these situations, this decreased mitotic spindle development. And if we look at individual genes that are most involved in that description, that they also decrease at the 15 and 1/2 hour points with the ALA study design. And so one of the reasons that heme contributes to a bigger cell is through presumably or potentially making them divide less frequently, and so they end up larger. And in addition, and not shown here, there are pathways that are specific to the RPL11 mouse, haploinsufficient mouse, and not the FLVCR deleted or control mice that reflect P53 activation. So this model also allowed us to dissect what was secondary to P53 and what was secondary to heme because of the comparator to the FLVCR deleted mouse. And so for the last bit of time, I wanted to share some unpublished data. And that is our single cell studies of DBA and del5Q MDS patients. And the experimental design is actually rather similar to the mice, except in this setting, and I'm happy to discuss why, we've used a 10x platform as opposed to the Fluidigm platform. And we also use the CIHD, which we had done before, as a method for quantitating cell surface protein. So the experimental design is to get lineage negative marrow cells, to barcode with and CIHD to quantitate the cell surface proteins, to then identify total transcriptomes with 10x genomics. We analyze cells directly and after 3 and 6 days of culture with the erythroid expansion. And interestingly, after 3 and 6 days of culture, the major differences were the ones that were published, which was the increased expression of globin, increased expression of transferrin receptor, but very minimal other differences on analyses of principal components. And that allowed us to combine these data. And in culture, these aberrant cells, these DBA cells and del5Q MDS cells differentiate further so that we could query what was going on with them because they were there. And then we assessed the transcriptomes with SC-Velo pseudotime analysis. And this was a really critical step. It includes incompletely processed RNAs. These are actually very prevalent and regulated during erythropoiesis. John Conboy knows that well. And our work was nicely correlative with his. And doing this actually allowed us to align results from multiple studies. So you can use single cell technologies that are well published and look at an individual study with a control patient and get wonderful results. But when you do that with the next patient, the results are disparate. And so if you want to combine erythropoiesis across many different patients, a critical methodologic thing for us was to align cells by their incomplete messaging. And so first to look at the proteins and whether they actually did a good job at expressing cell stage as it relates to gene expression. One used these standard protein definitions. So we looked with barcoding for the presence of c-KIT, CD71, CD36, and glycophorin A, and they differentiated across the traditional velocity pseudotime, very appropriately defining stages listed below by traditional criteria. Two-dimensional force maps are a way to look at how cells differentiate. And I'm going to pause for a minute here because this is really in some ways the most important and interesting part of our study. That if one looks at this force map, so this is defining cells by their relative changes in transcriptome. The erythroid cells differentiate across the stages A, B, C, D, G, F that were defined before with this color code. And so the stage C here is the CFUE, early and late CFUE. And very interestingly, that there appear to be two trajectories. we're going to call it trajectory B, and the normal trajectory, trajectory A. So some cells went all the way through differentiation, and some cells, the differentiation was aborted. And if one looked at these cells here, they had a very low ratio of a very less transcripts, so they had more mitochondrial transcripts preserved. And by many criteria, they were dead and dying. And so at the CFUE stage, just where heme is first intensified, cells are one or two trajectories, one that's healthy and one that's destined to die. And yet they don't die to a fair bit later. And if one looks at cell source over here, there's 73% of DBA cells that follow trajectory B. And this data that I'm showing is just the DBA data. The MDS data is actually quite similar. And that while most normal cells follow the normal trajectory, but interestingly, normal cells are also on both trajectories and DBA cells are on both trajectories, which allows us to ask questions about what allows a DBA cell that's abnormal genetically to mature normally. Why does a normal cell that's normal genetically have an abnormal ineffective maturation to it? And one can look at dot plots of differentially regulated genes, which confirm that trajectory B is a death pathway, and that the most upregulated genes in trajectory A were mitochondrial oxidative metabolism genes and erythroid genes, and the most altered genes in trajectory B were those that we've identified previously as associated with heme or heme toxicities. So it appeared that heme was helping the cell make a decision at this stage, normal cells and DBA cells, whether to have a healthy differentiation ahead or whether to die. So in broad summary of this, by studying the fates of single erythroid cells from DBA and del5q and controls, we're able to identify an unhealthy versus healthy, a pseudotime trajectory. The pathways diverge right after the upregulation of CD71 and the start of heme synthesis, although differentiation stops and cell death occurs quite a bit later, meaning that there's actually a time interval that one could potentially interfere thinking about therapy. That the cells destined to die express high levels of ribosomal protein and heme-regulated genes. And in contrast, surviving cells downregulate heme synthesis while upregulating mitochondrial oxidative phosphorylation, again suggesting perhaps new targets for therapy. And quite unexpectedly, I'd say 7 to 16% of control cells followed the unhealthy trajectory that implied to us that heme may also be a rheostat that kind of regulates normal cell production. And that perhaps one reason that there's so many excess or ability to respond to stress and to expand erythropoiesis is that these cells that heme would normally make die under stress situations are allowed to live and then contribute to expansion at this early stage. That's entirely conjecture, but one way to explain why so many control cells are actually dying. And then it also led to some additional questions, which are ongoing questions in the lab, which is that del5q MDS, unlike DBA, results in somatic mutation. So there's many normal cells as well as abnormal erythroid cells. And in fact, one of the confusions about why anemia develops and macrocytic anemia particularly develops in del5q MDS is that a presentation, a patient might just have 50% cells that are from that clone that are lacking chromosome 5q. And yet those normal cells can't expand and take over and support erythropoiesis. And so they're impacted by the presence of the del5q minus cells. And that we thought that tracking the trajectory of these cells individually might prove new insights to why they couldn't compensate and why they can't expand and prevent anemia. And so just as some words for some preliminary studies of looking at the pathways of the normal, genetically normal cells in a del5q MDS patient, that is the 5q cells that are in trajectory A, the healthy trajectory, we found that they're not entirely normal, that their transcriptomes actually differ from the transcriptomes of normal trajectory A cells and control subjects that were studied concurrently. And actually, interestingly, some of the differences relate to heme. And so it, as a preliminary data, suggests that the MDS patient cells with a del5q deletion directly or perhaps indirectly, maybe by impacting the surrounding microenvironment, impact the effective differentiation of the neighboring normal genetic cells, genetically normal cells. And then as a last finding, that interestingly, the oxidative phosphorylation RNA splicing cell cycle checkpoints in HIF1 alpha pathways are altered, and that these are similar findings to the patient's del5q cells, the cells that are actually lacking the 5q, suggesting that they too may have anti-DBA patients, suggesting that this might be a heme-related process, at least in part. So back to Diane's question. From medical school onwards, and probably in graduate school onwards, we talk about red cells as being enucleate sacs that carry oxygen, that carry around hemoglobin to transport oxygen and to pick up CO2. And they're normocytic, meaning they have a normal MCV, which is 80 to 100. They're normochromic, which means that their mean hemoglobin concentration is normal. And their, the mean hemoglobin content is normal. And their MCHC is normal, their mean hemoglobin concentration. And the mean hemoglobin concentration is critical to be normal because that's what allows for optimal oxygen delivery and CO2 return. And so there's probably billions of mechanisms, given evolution, that control cell size as it relates to hemoglobin to assure that MCHC stays the same. And that means that oxygen delivery is fine and CO2 return is fine. So in iron deficiency, characterized as shown on the right, there is a small cell size, it's microcytic. It's hypochromic, so its content of heme totally is low. And this happens when you can't make either enough hemoglobin because of failed heme production, and iron deficiency would be an example, or failed globin production, where thalassemia is an example. And cells try to maintain a normal MCHC by making smaller cells. So therefore they can function appropriately. And there's many mechanisms, including HRI, that have been described that probably connect this way. So the thought that I want to leave you with is what's happening in macrocytosis. That is cells that are big, that have high hemoglobin content, that have done so potentially to maintain their MCHC. And so when there is abundant or excess heme, are cells made larger as a compensation to try to maintain MCHC? And what might be the different mechanisms for that? Ones where heme is in excess might be heme regulated to compensate. Ones where there is a intrinsic problem or intrinsic excess of the cell size might then induce heme to compensate. And our thought is that this is going to be a consistent finding really across a very simple finding to link cell size and the things we're used to looking at as metapathologists and cells. And so with that, I'd like to thank my group. This is a very old picture of a lab hike to Mount Rainier. And I picked it only because the studies that I'm showing really have gone on over much of my lifetime and have evolved over that time. And I'm particularly thankful to Raymond Doty, who has been responsible with Chris Losted for most of the recent single cell work. Raymond did most of the mouse work, and Santal Yang was responsible for most of the earlier human work, and for our many foundations and a very long-standing NIH grant that funds these projects. And so thank you, and I'm happy to answer many more questions.
Thank you, Jan, very much for the presentation. We have several questions in the chat. I'm going to start by inviting Tom Gans to unmute himself and ask his question.
Yes, nice talk, Jan. Have you tried iron restricting the cells in vitro to see if the bifurcations that you're seeing, both in normal cells and abnormal cells, can be modified by iron restriction?
Proposed in our R01. Yes, we haven't done it yet, but that's the you know, to repeat these studies. Single cell studies are actually quite expensive still, so we need to figure out the optimal design. But we've used iron restriction, we've used succinylacetone, we've used several different methodologies in our cultures before with bulk cultures, and we probably will repeat, probably in our restriction way it's simplest. And the way that we do it simplest is change the concentration of APO versus full transference, so we can do it with a lot of rigor. And that's the experimental design that we plan ahead. We know that the cells will grow better, and the question is how will they distribute, how will they compensate for that? And the interesting thing for the del5Q is, well, the interesting things with DBA in general is it also will allow some separation of heme iron and P53 pathways. And it also, we think, will build a platform to look at other MDSs and other macrocytic anemias when we have that full data set.
Thank you.
Great. Let's invite Sarah L. Haas to ask her question now.
Very interesting talk. Thank you so much for this. I wanted to ask you about the ferroptosis in the differentiation. Did you specifically check for ferroptosis and apoptosis and try to differentiate them? And how do you think the erythroblastic niche can affect the differentiation? Did you try and look at that?
Okay, so a nice and loaded question. The answer to the first part, if we look at to separate ferroptosis and apoptosis, we didn't. In our earlier studies, the bulk studies, we looked at numbers of the gene expression patterns that are changing with both and saw both present. And so I think both are present. And as more emerges about ferroptosis in many systems, I think ours is probably one where ferroptosis is quite prominent, but we haven't done a lot of work there. With regards to your second question, I anticipated it. And this is another direction of the lab that I really can't give a lot of data about yet because we don't have that data yet. But our hypothesis is that what we're trying to figure out is why are normal cells affected in an individual that has a combination of cells lacking chromosome 5q and cells where chromosome 5q is intact. And why that's particularly interesting to us is that if we look at mice that are FLVCR deficient and transplant them 50-50 into normal mice, erythropoiesis is normal. So that FLVCR doesn't inhibit, FLVCR deletion does not inhibit adjacent cells. And actually, if we do that same experiment with 5% normal cells and 95% FLVCR deficient cells, those normal cells expand and erythropoiesis is normal. So if one can't export heme, then you can compensate. And if you export excess heme, you can't. And that's led us to think about erythroblastic islands. And these, again, are just early studies. But most erythropoiesis in the marrow, about 85%, old work actually of Mohan, takes place in erythropoietic islands, within erythroblastic islands. And a single macrophage, it's often termed a nurse cell, can have 50 to 100 or more erythroid cells on it. One of the sort of oddities about erythroid islands is that erythroid cells at all stages of their differentiation adhere. And so if the purpose of the erythroid island was just a phagocytosis very late cell, then it's really unclear why CFUE and proerythroblasts adhere. And this led us to think that maybe erythroblastic islands are very efficient heme iron recyclers. Heme and iron are toxic. It's going to, if proerythroblasts are 50 or 100 attached to a single macrophage just exported into the environment, there might be toxicity in the marrow. So perhaps it goes directly into macrophages who then make it into iron. And very late in erythropoiesis, cells are actually quite iron avid. And so that perhaps that actually expands erythropoiesis on this side. And so we're working with trying to work this design in some in vitro studies. And the thought process would be that if adjacent CFUE that were del5Q positive and del5Q negative were here, and they're exporting lots and lots of heme now into the macrophage, that it may make the function of this macrophage aberrant, and therefore impact the normal neighboring cells that are genetically normal, and that this is the impact. It may even impair the ability of the normal or neighboring cells to export their heme. Because one thing we know about the biophysical characteristic of FLVCR, which is the only heme exporter, is it requires something to receive it on the other side to function. So this could be hemopexin, it can be hemalbumin, it could perhaps be other things, but it has to be something specific, or else the cell dies, it doesn't export, it's unable to export heme. And so we're sort of exploring this physiology as a way of thinking of how the external influence could be, how there could be a cell extrinsic factor that impairs the differentiation of the 5q plus cells present in the patient with del(5q) MDS.
Great. A couple more questions. One from, I'm going to apologize if I mispronounce this, Katarzina. Do you want to unmute yourself?
Thank you. Yes. I'm wondering about heme oxygenase 1. So it was reported by Prem Komponka to be expressed in erythroid cells during differentiation. So why heme accumulation does not induce the enzyme in cells derived from DBA patients, and if HO1 could play any role in detoxification of heme?
Okay, great question. So, so Prem's work was actually in fetal erythropoiesis, which is a very different situation. Actually, heme oxygenase is not expressed if we look at it across human adult erythropoiesis. And so it kind of led us to think about why would it maybe not be expressed, just broadly thinking. And here's the thought process that very quickly globin is made and induced by heme. And if heme turned on heme oxygenase 1, the protein actually has a reasonably long lifespan, and it would complete with globin. So it wouldn't really be in a red cell's interest to control heme that way, because a few seconds or a few minutes, we don't know the exact time course later, it needs all its heme because it has globin that's present. And so exporting heme is a safer way to preserve the production of heme so that it's ready and able whenever globin is there. And there's also a second reason that crossed our mind, and that is in the marrow, erythropoiesis takes place in a relatively hypoxic part of marrow. And so heme oxygenase requires oxygen. So as a protein, it may not be particularly functional. And so for many reasons, we think that that's not the case, but we're certainly obviously interested in overexpressing it in the macrophage as a way of testing hypothesis.
Thank you. Clara, do you want to unmute yourself and ask your questions?
Yes, thank you. Great talk, Jan. Thank you very much. I was always fascinated by this story of the size of red cells, small size or large size, microcytic and macrocytic. And so trying to accommodate your model in all types of anemia, I was thinking of forms of congenital sideroblastic anemia that obviously they have defect on heme and so are microcytic. But the acquired form that you mentioned, MDS-RS with ring sideroblast are macrocytic. So what can you think about that?
Yeah, we're actually thinking a lot about that. So as Clara says, the acquired forms are often from abnormalities, for example, the X-linked sideroblastic anemia from an abnormality of ALS2. So you don't make heme. So it's obviously would be microcytic because you don't, for the same reason that other cells are microcytic. Yeah. So the real question is why are RARS cells, yes, with ring sideroblast macrocytic? And I'm going to have to let you wait for some work by my colleague Sergei Dulatov, who may be on the call, to explain that. But the thought would be there is going to be some, that whatever is going on there is not sacrificing the ability to actually make heme. That has to do with the fact that iron sulfur complexes control some of the genes on the heme synthetic pathway, as suggested by Tracy. For example, a RUALT or another mechanism is really has to be figured out. But we appreciated that disconnect, and it's driven some of the work in my collaborators' lab.
Okay, thank you.
Thank you. One more question. Last question by Bruno Gali. Bruno, would you like to go ahead and unmute yourself?
Yes, it's a very general question. There is a lot of ribosome diversity as shown by the lab of Barna. There is a kind of ribosome code, like there is a histone code. And I was wondering whether there is anything known about changes in ribosome composition or post-translational modifications beside the changes in expression that you see in erythroid cells. Did anyone look into that?
I'm not, I'm happy for anyone on the call to answer if that's something that they know about. I really know nothing about that topic.
I don't hear anyone chiming in. So Jan, we want to thank you for the great presentation today, really actually thought-provoking. As a cell biologist, I'm trying to think about, do you see those macrocytic cells be big early? Like, do they still have, and then that just retains that? I'm trying to think about where their membrane is coming from.
Yeah. I don't know the answer to that. It's been studied predominantly late in circulation. I'm sure it's knowable where they might become bigger early. I just don't know that answer.
That's okay. Thanks so much. And want to thank everybody for their attendance. I'm going to just launch a quick poll if you're still on. This is my first time doing this, so we'll see how it goes. And this is the first question I'm going to launch. And then just if you work through the questions, we'll know if we want to do these seminar series again in the future. So just feel free to kind of move through them and answer them. And we'll just take just a couple minutes. And I'll just ask, can you see the poll?
Yes.
Awesome.
It doesn't appear to submit, however.
You
I
know,
think you have to answer all the questions first.
yeah.
Oh,
Yes,
I'm
hold on.
sorry. Yeah, okay. Oh, sorry.
That's okay. Thanks for the question. I'm like, oh no.
Okay.
It's a great participation. Thank you so much for those who are doing it. You can give me a thumbs up if you're done and we can end the poll at some point. I don't see it accumulating. A couple more.
Diane, there is one problem with the poll. It asks us if we want to schedule to be more frequent or less frequent. Some of us don't want any change at all, and you didn't give us that option.
Okay, I was too binary.
Yes, too binary.
Okay, thank you for that. Thank you.
Okay.
So about once a month seems like a good idea.
It seems like a great idea. This is a good rhythm.
Okay, I really appreciate it. Thank you so
I
much.
clicked on one of them, but it's irrelevant because I'd like no change.
Okay, I appreciate the comment very, very much. Thank you so much, everyone. I'm going to end the poll now. And I just want to remind you that if you're interested, the Centers of Excellence in Molecular Hematology, sponsored by NIDDK, also has funding opportunities where you can get you can apply to get funds to use the core facilities that are available at the different centers of excellence across the United States. And I highly recommend you go look on their website, which is cceh.io, for that information. And again, thank you all for your participation. Take care. You can unmute yourselves and get yourselves on the video if you want for a second to say hi, everybody, because it's just so great to see you all. Really great.
Hi.
Bye. Bye-bye.
Bye.
Everyone.
Bye, everybody.
Nice
We'll
to see everybody.
reschedule.
Thank you, Diane. Thanks, Jane.
Thanks.
Thank you.
And
You're
next
welcome.
time in
Take care,
some nice
everyone.
place at the other end of the world.
Okay. Bye-bye.
Thanks. Bye,
Ciao.
everyone.
Ciao.
Bye-bye, everyone.
Ciao.
Thank you.
Ciao.
Great talk.
Bye.
Bye, everyone. Thank you so much. Thanks
Thank
again,
you.
Jan. Great talk.
Thank you, everybody. It's fun. It's great to see you all.
Let everybody pop off.